Well, thank you all very much for having me here tonight. I wasn't sure what to expect. I was expecting a smaller group, but it's super exciting to see so many of you here. And hopefully you're still excited about this topic after I'm done talking about it. All right. So don't stress about the test. The test is there if you want to follow along and see if you can answer the questions. And at the end, if I haven't answered those questions, then you should ask me to explain what the answers are. And can everybody hear me okay with this? Okay. All right. Please, if I start drifting away from the microphone, please just holler at me. All right. I'm going to start with an acknowledgement slide because what I'm going to talk about tonight is sort of a lot of work combined into a very short presentation relative to the amount of time it took to learn this information. And the people in the different groups, the top left there are collaborators that I work with. Some of them are international. Staff down in the bottom left group, students, undergraduates, and graduate students on the right-hand column, and then agencies and organizations who have facilitated access to land for site access, and then countless landowners who have allowed us to sample on their property. Really, without private landowners in Texas, it's really hard to get much done. So thank you to all the landowners. And then the funding that has supported a lot of this work is from Texas Parks and Wildlife, a lot of it, the U.S. Fish and Wildlife Service, the City of Austin has funded some of this, and the U.S. Army Corps of Engineers has funded a bunch of this more recent work that we've been doing. And I'm not talking about specific projects, but because bits and pieces of this information came from all those projects, I really want to acknowledge all the supporters that we've had. And I also want to thank you for putting together this super cool announcement. I really like this. I sent her a whole bunch of photos in an album and said, here, I don't know what you're looking for, but you can pick. And this lovely announcement came out of it.
Sit back there, creators.
Okay, well, whoever did this, thank you. And I really like the fact that the Master Naturalist logo is a dragonfly. Some of you, I think, already know that I'm also fascinated by dragonflies. And so this photo was me in southeastern coastal swamps of Australia with a very rare dragonfly, in case you're wondering what I was pointing at. All right, so I wanted to start with a little bit of background about what I do, and that'll lead into the karst and hydrogeology part of this. So I am a hydrogeologist. I'm a karst hydrogeologist, and I'll explain what karst means if you don't already know what karst means. And when I got the job at Texas State University, I was hired as a karst hydrogeologist into a biology department. So a geoscientist going into a biology department seemed a little bit strange to a lot of people, but it was really exciting for me because it gave me the opportunity to do a lot of interdisciplinary research. And since that time, I have collaborated with biologists on all kinds of projects, ranging from wildlife to groundwater organisms. I didn't know much about groundwater organisms at all when I started working at Texas State University. So ecological studies, basic and applied biology, and then as part of this, as you'll see as I go through the talk, taxonomy, which is the process of formally describing new species. And so there aren't many people that do that for groundwater invertebrates or critters. And so we've kind of taught ourselves how to do this over the past 10 years. And me and my collaborators. So I do want to say a couple things about the top two images here. The right-hand image is field sites from part of the Army Corps project that I mentioned. And that project has really focused on something called the hyporheic zone. And I'm not going to spend a lot of time talking about it, but it's the interface between groundwater and surface water that lies beneath streams. So you have an exchange of water and nutrients, and the organisms, the benthic organisms, can move down in the sediments and interact with groundwater organisms, and groundwater organisms can move up and interact with the benthic organisms. And it's a zone that we know relatively little about compared to the benthic zone. And all those little dots over there are places where we've sampled. And we've learned a huge amount about the distribution and range of groundwater organisms in Texas from this hypereal sampling. So that's why I wanted to point that out. And then on the bottom is just an example of some of the things that we worked on with some snail taxonomy, describing new species of groundwater snails. All right, so these were the quiz questions. If you're in the online audience, you could do a screenshot of this or take a phone photo of this, or just ignore it if you prefer. No stress. And if you see things on this list at the end that I haven't answered, again, please raise your hand and challenge me to answer them. Okay, so the structure is kind of like this. The first 20 minutes or so will be about karst and geology, hydrogeology, a little bit of basic geochemistry and hydrogeology, and then working into caves. Not really in depth on any of this, so if you want specifics, feel free to ask questions at the end. And then that will sort of set the stage for the habitats, the caves and karst conduits for most of the subterranean biology topics that I want to talk about in the second portion. Excuse me. All right, so I want to start with basics because some of you, I assume most of you know what an aquifer is in some general terms, but from a hydrogeology perspective, an aquifer is some geologic formation that produces usable amounts of water. And whether something is classified as an aquifer is really dependent on the user. So a large municipal pumper is going to define an aquifer very differently from a homeowner who just needs a small amount of water from a well. There are three general kinds of aquifers in hydrogeology. There's the porous media, which is the aquifer we're sitting on right now, the Gulf Coast aquifer, which is mostly sands. And water is moving through the interstitial spaces between those sand grains kind of slowly and relatively uniformly through the sands. There are fractured rock aquifers. Those obviously occur in places where there's hard rock. Not much hard rock here, but if you go to, I don't know, like the Llano Uplift or the Rocky Mountains, there's lots of rock that the water is moving primarily through the fractures. And then there's karst. And karst is kind of the opposite end of a spectrum, ranging from the porous media, where things are moving slowly and kind of uniformly, to karst, where things are moving quickly through conduits or caves. This is another word. If you've ever been in a cave, you were in a conduit. And so the images down on the bottom right are just illustrating that. And then there's a cave entrance looking out of a conduit. Karst is found all over the world, and the term karst comes from an area in Slovenia, but it is a term that's used to describe a landscape that forms in soluble rocks. And the most common soluble rock that karst forms in is carbonate, limestones and dolostones. And we have a lot of that in central and western Texas. Also in gypsum, we have some of that in kind of northern Texas and western Texas as well. And then other rocks like sandstones and salts around the world also form karst systems. Basically, if a rock is soluble, you will have the formation of some type of conduit, large or small, in the subsurface that is acting as a pathway for water to move through. So one simple conceptual way to think about karst systems is they are defined by these preferential flow paths, which lead to enhanced subterranean dissolution of the rock. So you're converting the solid rock into dissolved minerals in the water, and then that's getting transported through the conduits to a spring or out into a river and transported away from the system. So it's this integrated flow and mass transport system. And here's a schematic diagram that pretty well illustrates the karst that we have in central Texas. So most of the limestones in central Texas are relatively flat-lying, Cretaceous-aged carbonates. If you look at the top of that figure, you can see a little mountain. Maybe you can imagine that's the Llano Uplift, which is granite. It's up in the central part of the hill country. And you have a scattering of canyons. You know, the hill country has a lot of river canyons that are carved through it. You have some cave entrances and sinkholes. And these all are characteristics of karst that you would see on the surface. They're also all features that are created by that subterranean movement and dissolution of the rock itself. So without the conduits in the subsurface, you don't have those sinkholes happening, and you don't have caves that you can enter. So where does karst occur in the US? It's all over the world. It's on all continents in different amounts. And in the US, we have three primary types. This is a map that was put out by the USGS, and it's the karst map. And you can see in central Texas there's a lot of blue. That's all limestones in the Edwards Plateau and Hill Country and the Edwards Aquifer region. The green is gypsum, places where there are gypsum karst regions. So you can see it in kind of North and West Texas, we have some. And then in the far Northwest United States, we have those pink areas. Those are volcanic karst. They are primarily lava flows where they leave lava tubes behind. So that's it has tunnels and conduits, but they're not dissolved out of the rock like the rest of these systems that I'm talking about are. But they have similar habitats in terms of those conduits for organisms to live in. Zooming in, this is a map from the Texas Water Development Board. And this is a map of major aquifers. There's also a map that shows minor aquifers in the state of Texas. And we're down on the bottom right there along the coast in that Gulf Coast aquifer. So quiz question, answer. And then moving to the northwest, you can see that red band, and then there's a hatched area down to the right of it. That's the Carrizo-Wilcox aquifer, which is also a sand aquifer, very thick sand and some gravels. And then moving farther to the northwest, you see that blue curve, the arc. That is the outcrop of the Edwards Aquifer, the recharge zone for the Edwards Aquifer. So that's all Cretaceous limestone. And that's where San Marcos and Austin and San Antonio, those cities that were built around the big springs that come out of the Edwards Aquifer are. Then both colors of the green are the Hill Country and the Edwards Plateau aquifers. And those are all three, the greens and the blue, are basically one connected system with smaller internal drainage areas. And then to the north is the Ogallala and a couple of the other major aquifers that are largely porous media sedimentary aquifers, sands and gravels. In the hill country, if you walk around on karst, you'll see things like the Edwards limestone. There's a chunk of it on the top left. Oftentimes, the pieces of rock are full of little holes. Those holes have been dissolved out by the processes that are dissolving the rock. And if you imagine a whole block of rock, a whole hill country full of little holes like that, you can imagine there are thousands or maybe millions of miles of small little passageways that invertebrates can move through and live in. Humans, we can access caves like in the top right there. And most of the time, I'm not a cave diver, but sometimes people go underwater in caves. But most people access caves that are air-filled. Those caves were once water-filled. So through the erosion of the landscape, the water levels have dropped in the aquifer and exposed those caves, or drained those caves rather. And that drainage leads to features like on the bottom left, which are large springs in this case, at Comal Springs in New Braunfels. This is along that I-35 corridor. It's the largest spring in Texas. So that opening is one of several openings. How many of you here have been to New Braunfels before? Anybody? Oh, wow, a lot of you. So you've probably seen this spring. Spring run number one. Yeah, Land Park, exactly. So spring run number one normally looks like it does on the left there. On the right, that's my son standing in the spring run number one last year. We're in the middle of a 4-year drought, and the combination of drought conditions combined with groundwater pumping have led to the drying up of most of those spring runs. There's still water in the spring, but it's much, much below normal flow. Okay, So again, the hydrogeology here, I want to just emphasize the differences between karst hydrogeology and non-karst. And so at the surface, remember these features like sinkholes, springs, and caves are the visible features on the surface that indicate these subterranean conduit systems that have formed in a soluble rock. Here's a quick comparison table that leads into, we'll talk about the habitat differences in a second, but in karst systems, again, we have these conduits that allow rapid flow and movement of the water and anything it's carrying through the system. So you have turbulent flow conditions like you would in a river or a stream. In the non-karst, like our aquifer right underneath our feet, the water is moving very slowly through those pore spaces in what's called laminar flow conditions. And that's through the intergranular porosity. Sometimes there are fractures in hardened sandstone and things like that. So the high velocity versus low velocity. And the karst systems comparatively are extremely heterogeneous, meaning they have different properties through space relative to the porous media aquifer, which is relatively homogeneous across the aquifer system. And from an animal perspective, what this does is it creates really different conditions between these two systems, and in the karst system, it creates really different conditions throughout the aquifer. So differences in size of the openings, so that will control how large an animal can move through them. So some critters can move really well through very tiny openings. Some of them have to have bigger openings. You'll see some of the critters later. The amount of dissolved oxygen that's present, dissolved and particulate nutrients that are carried into the aquifer will vary depending on the properties of the aquifer, and then the environmental stability. So something like a sand aquifer, the stability of the conditions in the aquifer will generally be pretty consistent through time, especially if you're deeper in the aquifer. In a karst conduit, this is data collected from a stream in a cave in Virginia. Each one of those little spikes, it looks like you're looking at a river on the surface, a flow gauge on the river. And that's exactly what's happening. There's small rain events, and that stream just changes hugely in terms of the flow that goes through that conduit over very short periods of time. Okay, so think about karst as this integrated system, and I'm going to use it evolves into this integrated connected system that transports water and dissolved solutes. And I'm using the term not in a biological sense here, but in terms of like positive feedback loops, where the more it dissolves, the more water it can carry through it. And karst scientists call this, in really simple words, an integrated mass transport system. It's a fancy way of saying it's a pipe carrying stuff through it. Yeah. And the stuff that is getting carried through it is that dissolved rock. So we see beautiful, clear spring water. There's actually a huge amount of dissolved mineral load that comes through that spring every day that was solid rock at one time. So this is sort of chemical erosion happening. And then also the sediments and organic material in the conduits. In caves, you can move boulders through the conduits. You don't do that in sandstone aquifers, but in caves and streams, you can move very large particles through some systems. And I've seen tree trunks like log jams in caves. So lots and lots of organic material can go into some caves. All right, I'm not going to spend a ton of time on geochemistry because it does get super complicated, but I'm going to talk about the very basic part, which is, if you remember, rain falls through the atmosphere, it picks up CO2, forms carbonic acid, and then it interacts with the rock, the carbonate rock, and dissolves it. That's a sort of accurate picture. It does do that, but the amount of CO2 in the atmosphere is very small compared to the CO2 in the soil. Soil concentrations are hundreds of times higher than they are in the atmosphere. So atmospheric is around 4,000 or 400 rather parts per million right now. Soil CO2 can get 50,000 to 100,000 parts per million. So as the water lands as precipitation on the surface of the ground and infiltrates and percolates downwards, it's absorbing that CO2 in the soil profile, and then it moves downwards and it interacts with the rock. And so you have a much stronger, it's not going to burn you or anything, but compared to the atmospheric CO2, much stronger solution of carbonic acid that then interacts with the rock. And this is a construction site that I took some photos of. There's a bedrock pillar there. And if you zoom in on the right here, you can see in this zone that's called the epikarst, kind of the interaction or interface between soils and bedrock, that as the water moved down through along that rock, it started dissolving out these small conduits. So even very close to the surface in the soil horizon, karst systems start developing these initial conduits at that bedrock soil interface. And the epikarst is also the zone where you start regulating a lot of different processes, not just the hydrologic and geochemical, but these biological processes and mechanisms that then feed into what's happening down in the proper aquifer. And eventually you do get to a proper aquifer. So this is a cave in the Yucatan, Mexico, Yucatan Peninsula. This is the water table in a cave, a long passageway that is draining water from the interior of the peninsula out to springs along the coast. And as the water comes down, it enters that aquifer and is full of minerals, dissolved minerals. Some of that water, as it comes through the ceiling, though, you'll see these stalactites and stalagmites. When it comes out of the bedrock in the ceiling into the cave atmosphere, which has back to basically atmospheric concentrations of CO2, the CO2 that's in that water wants to escape, kind of like opening a can of soda. It doesn't literally fizz, but it's the same concept. And the CO2 escapes and leaves the mineral back behind, which precipitates out and forms those little incremental growths on the stalactites and stalagmites. All right. So the caves are one large component of the subterranean habitat in a karst aquifer. Those are the parts that we can walk around in. We can observe them. I like this photo because this photo, you can see those features on the ceiling, sort of scallops, we call them. And that tells us that that cave conduit, that passageway, was once filled with water, and it was moving pretty slowly. And we can crawl around in these or walk around in them, and we can find little invertebrates in them and study those and do all kinds of things in caves. But remember that off the sides are all these small little conduits, like in that chunk of Edwards limestone, that is a huge network of habitat for all the other tiny critters other than us. Okay, so this initial part, the main things I wanted to, and I hopefully communicated, were that karst systems form insoluble bedrocks. And that's not just limestones, but that is the most common bedrock that forms karst systems. That karst hydrogeology is characterized by this heterogeneity, rapid flow. So that's bad if you're talking about contaminant transport. Contaminants can very rapidly get into the aquifer system. And it is really this integrated mass transport system that's moving dissolved and particulate matter through it. The CO2 is mostly coming from the soils, even though rainwater does pick up some CO2. When you're walking around on the surface and you see things like sinkholes or cave entrances, that means there is an extensive network of conduits beneath there somewhere. We may not be able to explore them, but they don't stop when the caver can no longer continue moving through the cave. They continue hydrologically. And these are habitat, even if they're not humanly accessible. All right. So I don't know whether we want to just wait until the end to ask questions, or if anybody has any aquifer related questions right now. I'm happy to keep going, or, oh, we got at least one. So what does the presence of clay do to different types of aquifers? Well, in the Edwards aquifer, there are clay-rich units, bedrock layers that overlie the confined part of the Edwards aquifer and keep that water from moving upwards until you drill a well through them. So clays can confine systems, and in the Gulf Coast system here, there are clay regions or layers in the aquifer here as well. So in a whole aquifer system, they often act as a confining unit, either above or below, to prevent the vertical movement of water through them. Yeah. Oh. Oh. We're good again? Okay. Yes. And then I'll get to you.
The circular one in the middle of Michigan.
Yes. So why was there a circular karst system in the middle of Michigan? So I should have mentioned that that map is a map of surface exposure of karstic rocks or near surface exposure. Yes, and so that circular pattern is the result of what's called a geologic basin. So the rocks are exposed at the surface around the edges, and then in the center, it's been depressed. Yeah, or it could be the opposite in some situations where it's a dome and then the middle has been eroded away. Yeah. And then one question over here.
Find out, but back on the map where you have all the different curves of their drainage around us. So why was that not on the map?
The map of where I had sampled? This one? Yes. So we didn't sample the Brazos, you're right. Yes. So this, I'm sorry, this is not showing all the river basins in Texas. So the question for online was, why didn't the Brazos show up here? The Brazos is super important, but it fills in one of those white gaps. We did not sample in the Brazos on that study. So this was just sites in other river basins. I should have made that clear. Thank you. Okay, we'll flip back forward now. Hopefully I'm not making anybody seasick here. All right. If you have any more questions, you can hold them till the end now. Okay, so now that you have a general idea of what an aquifer is and what a karst aquifer is and what kinds of habitat might be down there, I want to talk about subterranean biology. And this is the outline of the topics there. So I'll dive right in. It's a dark slide. So, subterranean biology, what lives down there? Yes, Gollum lives down there. Gollum is on his way to becoming a stegobiont. So, in subterranean ecosystems, there are two groups of organisms that scientists describe. One is troglobionts, and the other is stegobionts. Both of those groups have all these characters that I list on the right. So loss of eyes, loss of pigment, elongated appendages many times, but sometimes shorter. We'll talk about that later. Generally longer living and slow metabolism. We think low reproductive rates. They may or may not be more sensitive to environmental changes. They usually have smaller ranges and restricted habitats compared to many species on the surface and other adaptations. And I used a lot of may in that because we're looking into something that's very black and hard to observe, dark places. And you'll notice there's a tiny little dot down there on the left. The analogy that I use a lot of times is that we're looking through pinholes into a giant black box and we're trying to understand what's down there. So if you would imagine going out into the woods with a little piece of pipe and laying down in the middle of a forest and looking through that piece of pipe up into a tree at a tree branch and waiting until you saw a bird and you saw a cardinal fly over and you see a cardinal in that tree, you're like, there's a bird. But you can't really say much more about it. Well, you can say some things about it, but it's hard to say much more about it other than you saw that bird in that spot. If you take that pipe away, then you can see the whole forest and you can understand a lot more about the bird and where it lives and what it eats and what it interacts with and where it nests and all these things. We know almost nothing about those types of things for most stegobionts or troglobionts. So troglobionts are for things that are terrestrial underground. So things like millipedes and cave spiders and things like that walk around, or beetles. Stegobionts are things that live in the water. So they are obligate underground organisms that live in the aquifer. And that's mostly, well, that's entirely what I'm going to talk about now moving forward. So the other ones are very important, but that's not what I'm going to talk about tonight. So we look through these little pinholes, and these pinholes into this giant black box come in a couple of different forms. Sometimes we can go into a cave and we can find something in a cave stream and we can observe it there or sample it there. Sometimes we can go to Comal Springs and we can put a drift net on that spring and collect the organisms as they're getting accidentally flushed out of the aquifer. And we know that they came out of the aquifer. We don't know where they were living in the aquifer, or what they did there, how many of them are there necessarily, but we can capture them as they're on their way to being eaten by a bass, for example, because they don't survive very well in the surface environment. And then we have wells that are drilled deep into the Edwards Aquifer, for example, and pumped, or in the case of the Edwards, which is artesian flows in many places, they can flow out under natural artesian pressures, and we can put a drift net on those and sample them as well. So when you open up that tube and you look, you might see something like this. And we'll talk more about this little critter later, but this is a ostracod. And it's about 1/2 millimeter long, and you can see it's fairly translucent, has no eyes. Those little circular things are eggs, and you can see the little legs and some other seedy and like little hairs and things like that that it's using as it navigates through its environment. The diversity of things that live in the aquifer is, at least to me, is incredibly fascinating. The closer you look at things, I was talking to somebody about a good microscope before. When you look at things under a microscope, you see all sorts of details that you don't see if you're holding something that's 1/2 millimeter on your fingertip. You really can't see anything except that maybe there's something there. So this is showing you just a little sampling of some of the groundwater biodiversity that we know of in Texas so far. The salamander in the top left of the right-hand image is the Texas blind salamander, Eurecia rathbuni, and it is the first groundwater species that was described in Texas in 1896, and it came out of an artesian well that is about 50 feet from the back door of my building where I work. Yeah, so that well, and I'll talk more about this towards the end, is the most biodiverse groundwater site in the United States and one of the most biodiverse in the world. All right, so how do these critters live underground? That's a great question. This slide is a little bit complicated, but I'm going to gloss over the complications, and I'm hope that at the end of talking about this slide for a few minutes, that it will make sense to you. That's my goal for this slide. All right, so when you talk about food webs, there's a base to a food web, and then there's the top. So if you're in the ocean, you're starting with microbes, and you're going all the way up to the great white shark, right? And there are different ways of figuring out what lives in the ocean and who's eating who and where you are on the food web, right? This diagram is something called a stable isotope biplot. And you ignore that if you don't know what that already means. The numbers across the bottom are indicating the values of carbon isotope ratios. And terrestrial organic matter, so things that are derived from photosynthetic processes on the surface, have a signature that we can measure in samples. And those wind up over here where I have terrestrial organic matter on the plot, around 25, minus 25 value. So forget about isotopes and all that. Just think about minus 25 is terrestrial. Moving across the scale there, you can see something called chemolith autotrophy. Those are microbes that operate a little bit differently. They are rock-eating microbes, and they occur in many aquifer systems around the world, including the Edwards Aquifer. And because they use different pathways and different geochemical conditions, they create carbon that is derived, they create organic carbon that is derived from inorganic carbon that is part of the limestone. So their primary production is happening with the rock, not with terrestrial organic matter coming in from the surface in particulate form or dissolved form. Like dissolved organic matter is something like tea and coffee, right? You make a cup of tea, you've just made a cup of dissolved organic matter. So these chemolith autotrophic food sources that are being generated by microbes, the biofilms, have a very different isotope signature. So just slide across there to -40 or -45. So how can we use this information to understand the food web in the aquifer? Well, we can go and we can collect the critters from the aquifer, and we can analyze those values, those isotope values, and we can then infer that something over here on the right-hand side, where that blue triangle is with SR in the middle, that those critters are probably eating more terrestrial organic sources, right? So the food that's feeding that part of the food chain is coming from nutrients that are washing in through sinkholes and sinking streams in the recharge zone. But then things that are over on the left side, where that left blue arrow is pointing up, those are critters that are primarily eating the chemolith autotrophic food. And so we can follow those carbon signatures through the food web and see who's eating what and where that food came from. So we think about caves and aquifers as being isolated from the surface, and sometimes they are. There's still transport of organic matter from the surface into the subsurface, but some aquifers are incredibly productive, biologically productive, because of these other conditions that occur. Okay, so I hope that made sense. Now let's go to the other side, the vertical axis, where we have numbers from 0 to 15. So these are representing the concept of trophic levels. So as something eats something else and something else eats that and something else eats that, you're going up in the food chain. And these are nitrogen isotopes. And again, that doesn't really matter, but what matters is that if I eat something, then the nitrogen isotopes are changed slightly during that assimilation, biological assimilation. And each time it goes from one level in the food chain to another, it incrementally goes upwards. And so this food chain at the time was the longest food chain in an aquifer that we knew about in the world. And it's almost the same as something that you would expect to find in an ocean. So it's very long, right? So down here on the bottom, we have microbes. And then up at the top, we have a great white shark. I see some heads nodding. Well, we don't have great white sharks in the aquifer. What does fit those spots, though? Okay, so here I'm going to just put some things onto that diagram and explain what actually what actually fits on that food chain. And it's really interesting because the biodiversity is fascinating. So at the top and the bottom here, I've put something called an amphipod, and some of you have probably heard the term amphipod. Some of you probably know what scuds are. So these are groundwater forms of those critters. And some of them are scrapers. So they're way down on the bottom of the food chain. They're just scraping biofilms and happy as can be, and then something comes along and eats them. Well, some other ones are way up here at the top pretending to be a great white shark, except they're less than ½ an inch long. The interesting thing here is that the largest organisms are not actually on the top of the food chain. So the salamander, they get several inches long, 4 or 5 inches, the biggest ones I've ever seen, usually about 3 to 4 inches long. They're not at the top. They're close to the top, but they're not at the very top of the food chain. These white things in the middle that look kind of like pill bugs, those are isopods, so they are related. They're in the same group as pill bugs in terrestrial environments. They get up to about 3/4 of an inch long. They're also not at the top of the food chain. Based on what we can see, they're probably just eating anything they can get. So they're eating biofilms, they're eating dead things, they're eating other things, they're just eating everything. So back on the bottom left, I put that little ostracod there again, and I put some question marks. So where does that go? Well, we don't know where that goes. And the reason for that is because for these isotope analyses, we need a certain amount of tissue. And those little ostracods, it would probably take hundreds or even a few thousand of these small ostracods to get that sample, and we just don't have that. But we can look at the type of legs it has, we can look at the mouth parts it has, and we can start making guesses about what it's doing in the aquifer, what it might be eating, how it's moving around, whether it has swimming legs or crawling legs. And the mouth parts on a lot of these organisms are really informative. So these are amphipod mouth parts, so back to the top of the food chain to the bottom again. And the ones on the top, those are the great white sharks. You can see they have big, chonkin, pointy teeth. They're out there swimming around, trying to catch other things and then tearing them apart, ripping them up with those sharp, pointy teeth. The things on the bottom are filter feeders or scrapers. They're eating biofilms and things like that. So mouth parts, even though they're not super exciting and we have to look at them with a scanning electron microscope, they tell us a lot about some organisms, especially when we look at them comparatively. All right. So where do groundwater critters live in Texas? That is a great question. And if you look at this map, you'll see there's a whole lot of red dots right around San Marcos and Austin. And there are several reasons for this. One of them is because that's where the first groundwater organisms were found in Texas. So the Texas blind salamander was described from a site at one of those red dots. And you'll notice that the dots get more sparse as you move away. That's because of observational biases. It takes 10 hours to drive to West Texas as opposed to 30 seconds to walk to the well outside the back of my office. So there are a lot of reasons for that. Another reason is that we are just starting to digitize this information. So a lot of the work that I'm talking about tonight, including this project, has been in really close collaboration with Ben Hutchins. Ben used to work for Texas Parks and Wildlife, and he came and he's working for me now. And he's putting together, he's kind of the mastermind behind the ABC, which is the Aquifer Biodiversity Database. And we have thousands and thousands of little jars with little critters in them. And we're trying to identify them and trying to enter this information into a database so that we can ask questions like where do they live and then provide an answer that's useful. So each of these dots represents a point where stigobionts or groundwater critters have been collected, and it's not a complete set yet, but we're working on it. So these kinds of data, from a practical perspective, are essential for any kind of conservation work. So if somebody comes to us and they said, you know, I don't know, we've had this where there's someone considering petitioning a species for listing as an endangered or threatened species. Well, you can't do that if you don't know where it occurs and where it doesn't occur, what its habitats are, how abundant it is, and things like that. So really, without a name and a location for a sample, that sample doesn't exist. And without a name on an individual name as a species name, it doesn't exist, unfortunately. So this is kind of where we started getting into taxonomy. We were discovering that so many of the things that we were looking at were undescribed species, and that's still true. We have a long way to go. So this is our extensive, it's very small compared to most museum collections, but we're very proud of it. It's about half of what we have, but it is now a formal collection. We uploaded the first 10,000 data points to GBIF just a couple months ago. So if you look up some of these groundwater critters in GBIF, you'll find records for them now, and they were not there for the past 130 years, not accessible. All right, I'm going to show you just a little bit of some of the weird places we find things. And I'll start off by just saying that everything you're seeing on all the critters you see on the slide are undescribed, meaning they have no scientific name at all. Some of them we're working on. So the ones on the left and the top right, these are species that we're currently in the process of working on describing. The three on the left all come from a single site in far west Texas. that previously we knew that that middle one, the isopod, that existed there, but it was an undescribed species that had been called something else. The top right one is really interesting. So we've been talking mostly about karst aquifers, but groundwater organisms actually live in all kinds of different environments. This tiny little worm-like crustacean lives in the sands in Salt Fork of the Red River in the panhandle of Texas. It's a groundwater fed stream. It's a really cool stream. And these are less than a millimeter long, so you can see that they're very skinny. You'll also notice that they have really short antennae, right? So I was talking about the elongated appendages. Species that are adapted to live in these interstitial habitats tend to have reduced antenna and reduced legs because they're worming their way through these sand grains or gravel grains. And then coming around to the bottom right, you'll notice there's no red dot at that point. That's because we don't have a sample from there. But this is an observation that was posted on iNaturalist several years back, and it's an isopod that appears to be a stigobionts. It kind of looks like it's lacking pigment, and it was reported as being in a small drain pipe on the Rice University campus in downtown Houston. So we have a lot of habitats that, back to those biases, people were biased towards looking at karst aquifers alone, and there's very likely a lot of groundwater biodiversity that's just been missed because we haven't been looking for it in these other groundwater habitats. All right, a little bit about the Edwards Aquifer, because most of what I'm going to talk about for the next few slides is all coming from the Edwards Aquifer, the Edwards Hill Country, or the Edwards Trinity Aquifer and the Edwards Plateau. The Edwards Aquifer is a karst aquifer in Central Texas. It's one of the most productive karst aquifers in the world. And you can see where it is in Central Texas. It looked like a lot of you have been to that area, so you know, you're familiar with this. Comal Springs and San Marcos Springs are the first and second largest springs in Texas. That red line is the jurisdictional boundary for the Edwards Aquifer Authority. The green is the contributing zone, so kind of a watershed. It is all karst in itself, but it's a watershed where water drains off the hill country down towards the recharge zone, which is in that greenish or bluish color band through the middle. And then the deep confined part of the Edwards Aquifer is in that tan color. And parts of that aquifer, wells are drilled as much as 3,000 feet deep to intersect that aquifer. So it's very deep, it's very difficult to access for sampling purposes. And it has some very interesting species in it, including the two blind catfish species that are known from deep wells around San Antonio. This is what that looks like in a conceptual diagram if you were to cut through it in a profile view. And you can see the contributing zone up on the left, the recharge zone kind of in the middle there, where it says Edwards Aquifer Recharge Zone, Balcones Escarpment. And the water then moves down into the confined part of the aquifer and then moves kind of in a northeasterly direction towards the springs. That bold dashed blue line there, that represents what's called the bad water line, or the freshwater saline water interface. So if you go deep enough in that aquifer, down dip towards the coastal plains, the aquifer transitions from fresh water that has oxygen into saline water that doesn't have oxygen. And that is that interface where those chemolith autotrophic microbes are just having a field day. They're just happy as can be down there. And they are literally eating away the rock. And I will make a statement that much of the permeability, much of the void spaces that have developed in the Edwards Aquifer are probably the result of microbes. So not necessarily the traditional carbon dioxide coming down from above and dissolving everything out. Those microbes are down there chomping away, and they've done this for tens of millions of years. And that's probably why we have such crazy biodiversity and such crazy permeability and huge productive wells in that zone. All right, so now we get to look at some cool pictures. What do these things look like? You've kind of seen a few of them, and I can't show you all of them. And I don't want to give the impression that there are thousands. Well, there might be thousands, but there aren't that many described. But we have some really crazy things. This is a tiny little isopod or amphipod, I'm sorry, that was discovered at Caroline Springs, which is a nature conservancy preserve, the Independence Creek Nature Conservancy Preserve out along the Pecos and Independence Creek, which flows into the Pecos River in West Texas. And this little critter is about 1.5 millimeters long, but look at the leg on that thing. Like, what in the world? So this is, well, it has 2 legs, but probably one of them got broken off before this photo. But this is a great example of something comes out of a spring, we catch it in a drift net, and we have no idea what that leg does. Like, why does it have a long leg? We don't know. We don't know where it lives in the aquifer. We know nothing about how many there are in the aquifer, in the habitats, where it actually lives. All these things we wish we had questions for, we don't. Here is one of the many amphipod species that comes out of the artesian well there in San Marcos. And this one is a great example of those elongated appendages. So it has relatively long legs, but it also has these super long antennae. The antennae are almost twice as long as the body. So from this, we can guess that this species, because it's almost a 1/2 inch long, the biggest ones, we can guess that it probably lives in larger conduits. It's not down in the little interstitial spaces worming its way between sand grains. It's swimming around, having a good time in the big old swimming pool down there. And we also only see this in the artesian well, which taps the deep confined part of the aquifer. So we suspect that it's living down there near that freshwater saline water interface. It's not found up in the hill country, at least that we've ever found it. So these habitats, the narrow ranges and small habitat areas are often what we see, these patterns. Here's another one this one was also described very early on from the artesian well in San Marcos. Stigobromus flagellatus. A lot of these have no common name. So, but you can see the flagellatus refers to the long flagellar antennae that are as long as the body are. And again, this one gets pretty good size. This is one of the biggest I think it is the biggest amphipod in the aquifer. And so we expect that it would be swimming around in the conduits. This one is about midway up on that food chain. So it's not the top predator, but it's not the bottom either. And here is just a panel of some of the other amphipods that we see in different aquifers, or different parts of the aquifer, rather, in the hill country or in the deep aquifer. And you can see sort of these ranges in lengths of antennae and legs and things like that. The one that's in the middle, in the bottom, that one comes out of, we often sample that out of gravels in the stream bed, so back to that hypereic zone. So if we go up into the limestone river gravels on the hill country and we sample the invertebrates there, we often get amphipods that look like this. They have shorter legs, shorter antennae, and it's probably because they're adapted for living in these larger interstitial spaces. Here are 3 different isopods of this group that kind of look like pill bugs. These are Cirlonides, and this is really the group that started us on this taxonomic journey of learning to describe species. The one in the very middle was discovered by my son when he was 9 years old. He was helping us sort in the lab, and he's like, hey, this one looks different. And I was like, yeah, sure. And I pulled out the description of it. And they're like, well, we'll compare it. I'll show you how this all works. Me being like the smart professor, right? And he was right. It was totally different. So the one that had been formally described is on the right. And I'm guessing that all of you can look at these 2 and see that they don't look the same. And they weren't the same. They're quite different. So those 2 are the only 2 species that are known in the aquifer right now. We're currently working on describing 2 more, and we think there are probably about 10 in total that we can put into different morphological groups. These are all pretty big critters. They get up to 1/2 inch or 3/4 of an inch, and they all live in these conduit environments. So these would not be little tiny interstitial critters. There are other groups of isopods. These are ones that unfortunately, when we sample them, all their legs break off, so they're really delicate. It's hard to get pictures of them. But these are ones that live in both interstitial spaces and in the deep aquifer. So Larceolus smithii, the one on the right, is only known from a couple sites right in San Marcos in the deep aquifer. The other ones are found in much wider ranges. And then this is kind of the extreme. If we're going from big chunky isopods to medium isopods to minute, those scale bars on the left are 1/2 of a millimeter. These are also isopods, and these are definitely things that you would expect to find in interstitial habitats. They come out of all kinds of habitats, though. So we think that even though they come out of the artesian well or we capture them at large springs, they're probably living in interstitial spaces somewhere in the aquifer, and then they're accidentally getting flushed out. So that the conduit is probably not where they want to be. But again, we don't know. So I put the blue photo up there because that one wound up on the intro slide. And they are not blue in real life. Yeah, no, they have their own little headlight system down there, but they have no eyes. Now, the blue, we were experimenting with some different microscopic methods for trying to describe these. And this is an image that was collected by a confocal microscope, which basically uses lasers with different wavelengths, and it fluoresces, and then you collect this image. And so you can see that that is also the same as the middle, or not the middle, but the one on the right. It's the same isopod, but these are transparent, whitish in real life. I put this panel on the right to kind of show you some of the things we have to do when we're describing new species. We have to dissect all these miniature little body parts and mount them on slides and then draw them and compare them to each other and all the other known species in that group in the world. And it's a slow and tedious, but also extremely rewarding process, because now we have 3 species that now have names, and we know at least some places where they occur. So another group, these are back to the ostracods. The top ones are 3 of the, I think we have 11 groundwater species that are known. And this is work that we've done with a collaborator from Turkey. So we have some species that have been named, but when we look at our samples, we see dozens of species that have not been named. So all these things on the bottom with these crazy spikes and these little wings, and the one on the middle right, I call that the bubble wrap one. Like these are our morphologies and characters that are really not known from groundwater ostracods. And then on the left and right are more simple ones. They're just smooth, or not just, they're still super cool. But none of these have names. So they're all in the large batch of things that we're waiting to someday describe and formally name. Back to the food chain for a minute. In the ocean, you have things like copepods and ostracods that are tiny little crustaceans that form part of the base of the food chain. We have those in the aquifers as well. So we have all kinds of different copepods. This is one group, harpacticoids. A lot of these things, I apologize, there just aren't really common names for them. So we could call them all shrimp, but that's not accurate because I'm going to show you a shrimp in a minute. But these are often extremely abundant. Like some samples that we collect will have thousands of these in them. And we don't even know how many species there are here because there are only a couple copepod species described in Texas from groundwater habitats, and they probably need to be redescribed because they were described a long time ago. So tons and tons of undescribed biodiversity here. Another group that doesn't have any names. These are these tiny little worm-like things called Bathynolasea. One of those on the middle right is that one that came from the Salt Fork of the Red River. So we find that when we sample the hyporheic zone, we find a lot of these interstitial species. We also find amphipods and isopods and some larger critters, but we find that many times these are relatively common in these interstitial habitats. In the past, these species or these critters were super rare in samples because they're small, so people would miss them. And also, they're probably not very abundant in the big conduits. They're hanging out in more protected areas, we think. Tons and tons of undescribed biodiversity here too. This is one of my favorite groups, the diving beetles. So I know if any of you have looked at samples from surface water, you're probably pretty familiar with diving beetles, the Dytiscids. And the one on the right is Hadioporus texanus. It was the first groundwater diving beetle, I think, described in North America. Came from the artesian well right behind my building. So you get the idea. This is a very biodiverse spot. And then all these other ones are undescribed species. And we have more. So this panel, I cut it in half. There's another. So these are sort of dorsal and ventral views of 5 different individuals. And so we're working with some folks right now to try and describe some additional species in these. Snails, work a lot with Dr. Catherine Perez from down at the UT Rio Grande Valley on groundwater snails. And again, just unbelievable numbers of undescribed species. We sat down not too long ago and we had found about 20 new species in our last year or so of sampling, and they're all still undescribed. But this gives you kind of a sense of the size. Many of them are very small, so less than a millimeter for a lot of them. And they come in all different bizarre shell morphologies. So that one at the top right is an uncoiled snail. I didn't even know such a thing exists. It's like a trumpet. Like it has this little tiny coil from when it was an egg, and then it just decides, hey, I'm not going to coil like a regular snail. And it just grows in a straight curved trumpet shape. There are 2 different species like this in Texas. Yeah, and just some really, so if you look at them under a scanning electron microscope, you see these incredibly beautiful, intricate patterns and textures on the shells. And sometimes these are used in the description. Some of them, like the top right one, don't really have any pattern or texture. So here's an actual cave shrimp. And I can call this one a shrimp because it is a shrimp. This is, as far as I know, these are the largest crustaceans that we have in the Edwards Aquifer. So they're the only things that are larger are the salamanders and the catfish, the blind catfish. These get about an inch long. There are 2 species of them. One of them is very common at the artesian well. They're only known from a couple sites in the San Marcos area and then a few sites in San Antonio. Maybe they're not even the same species. In San Marcos, there are 2 species, and they come out, they're very abundant, but the one species is about 10,000 to 1 of the other. So here's a great example of, is it rare, or is it just that the well is not in a place that accesses the habitat? The number of shrimp that come out for that one, surely there are more of them somewhere, right? But where are they? We don't know. The only point we can access that habitat is through the well or the spring nearby. So that's an example of where what we see at one site doesn't really tell us the whole story. It's tempting to say, oh, it's super rare because they only come out every 10,000 shrimp as this other species, but that is probably not the real story. I'm sure every one of you has had ticks on you before, right? And chiggers. Well, the same kinds of critters live in the water, including groundwater systems. I'm not saying they're going to bite you, but there are incredible numbers of aquatic mites, and many mites have an aquatic life stage, like parasitic mites that wind up on dragonflies, for example, have an aquatic life stage, and then they come out with the dragonfly when it emerges. Well, there are tons of mites that live in the aquifer, and their entire life cycle is happening in the aquifer. So the big one in the middle, you can see it has tiny little eye spots. This one is probably restricted to the hypereic zone. So it is not a true stegobiont because it doesn't have it has still got little tiny eyes there, is what I'm trying to say. The other 3 don't have any eyes. They don't have any pigment. I should say with the beetles, the color that you saw on the beetles there, that is not pigment. That is the color of the chitin that forms their exoskeleton. So they are pigmentless. They have lost the pigment. And if you compare those to diving beetles that you might see in a lake or something, many of those will be black or green or other colors. I'm going to end the picture show on this one. This is a really weird thing. It's called a gepigid, and it's only about 1.5 millimeters long, this one. They are incredibly diverse, but they're usually found in soils, in leaf litter, and sometimes in caves, often much bigger. But we also find them in drift samples coming out of springs sometimes, and we find them in our hyporheic samples. And based on some reading that I've done, some of these species may be sort of amphibious. So they might be living, if we sample the hyporheic zone beneath a gravel bar, for example, maybe they're living at that interface between the water and the air, and they're kind of going in and out of it. Some centipedes will do that, we know that. So perhaps this is kind of a species that is not a stigobiont or a troglobiont. It's a stigotroglobiont. I don't know. It's also undescribed. So you're kind of getting the sense that most of what we know about is unknown. All right. So what do we actually know? I've talked about a lot of things we don't know. What do we actually know about subterranean biology in Texas? We can plot things like this, which is a species accumulation over time. So as things have been described, the number of species increases. This is the artesian well behind the Freeman Aquatic Building on Texas State University campus. There are currently 40 species that are documented there that have names. And I would guess that if we described everything that we know as undescribed, it would probably be closer to 80. So even though this site has been studied since 1896, it still has a huge amount that we don't know about it. All right. So the total number currently of stigobionts in Texas in 2025 is 152 species. And you probably guess from everything I've been saying that it's actually way more than that, but we can't count them until they've been formally described through the taxonomic process. And I put the salamander there because that was the first one. It's also what we call the charismatic megafauna of the aquifer. So I meant to say this earlier, but if you ask somebody what lives in the Edwards Aquifer, most people in the San Antonio, Austin region will say, oh, the blind salamander. There were beers named after it. There are bars named after it. You know, everybody knows about the blind salamander. And if you ask somebody, what does the salamander eat? That's the response. No, yeah, beer. Nobody really knows because they don't know about all these other creatures. And that obviously is what the salamander eats. All right, so I've just switched to another plot here, and you can guess that I'm probably going to compare Texas to someplace else, right? So how does Texas compare to someplace else that also has karst, that has about the same land area as Texas does? France. We don't have the most current data, but France has about 354 named species as of our last counting there in the early 2000s. They also have a 100-year head start. So, so they started their first species around 1800. We started our first species count at around 1900. I'm sure that the species count in France is going to just continue and continue, and same in Texas. What's interesting here, though, is that if you were to sample and describe species in a terrestrial habitat, over time, you would expect this curve to be going the other way. Like as people study the system, fewer and fewer new things are found, right? You imagine what the species by year count looks like for birds in the United States. I don't know when the last new bird was described in the US, except for like splitting. You know, it's probably not been very recently, I would guess. I don't know. I'm not an ornithologist, so I'm sure one of you probably knows a lot more than I do. But these curves are going almost vertical now at the end, instead of going like this. And I know this is not the same exact data here, but this is kind of the shape of the curve that you would expect after a system has been well studied and all the species have been described. So just to make the point here that these systems all over the world, this is not just unique to Texas. This is true for groundwater organisms all over the world. Europe has a big head start on everybody. That's where biospeleology began, 100 or more years before it did in the United States. But all over the world, the same problem of many, many undescribed species. All right, so the bonus round that I didn't list on the first, I just want to quickly go through this. So some of the obstacles that we have to studying, understanding, and managing groundwater biodiversity, and maybe how you can help if you're interested. So a couple of the obstacles, taxonomic research is really poorly funded. Read basically non-existent in the United States. So I can't write a grant with any chance of success to go and describe a bunch of species that I know exist, as much as I would like to do that. So we're doing this basically in spare time and with spare funds and things like that. Texas access to land is difficult. So if we're trying to figure out where species occur, that private landowner interaction is essential. And that's where, perhaps some of you have springs, or of people with springs or wells with critters. We sometimes have private landowners say, hey, I have a spring. I saw some white critters in it. Maybe you do, I don't know. Maybe you haven't looked and now you will. Habitats are vanishing, especially groundwater dependent habitats. So many springs, especially in arid environments like far west Texas, these springs are drying up. There are thousands of springs across Texas that have dried up and will never flow again. And each one of these had some groundwater dependent ecosystem associated with it. And in arid systems, especially springs where they're not connected to rivers, the groundwater comes out, flows across the surface for a little bit, creates an aquatic patch of habitat. If that dries up, there's no other population to recolonize it. It's just gone. So this kind of thing is always happening. It's sometimes a little bit depressing, but we're just doing what we can. We have a lot of sampling biases. We know that. Access issues, aquifers versus hyporheic. I think we already know that. How many undescribed species exist in Texas? It's uncountable, so that was a quiz question. And then these are some things, ideas for how different people can help, like permission, support organizations and foundations that fund basic biological research or access for this sorts of thing. And there are often opportunities in places where there are museums and museum collections. There are opportunities for people to volunteer to help with that data curation. So we have thousands of little vials, little glass jars with things, and that information is really valuable, but it's not accessible right now. So it's just a slow grinding process of getting all that information put into the database. All right, some online resources. I think this is being recorded, so if somebody would like to look up some of the websites here for information about karst and karst biology, things like that, these are the sites. And there's my, go back. Sorry. Yeah, there you go. I'm not quite. I have to go back to my dragonfly. Everybody good with this slide? Yeah, I'm kidding. I'm ready for questions.